Hello, everyone. Today, I would like congratulations. I give it a congratulations for the organizers because this hybrid event for me it's so good. Stay here with you, and I know the hybrid events is not so easy to organize. And for this, I give a congratulations for the organizers. I think in Chennai, in India, is afternoon. In Brazil, I am in Brazil now, and here is around 9 o'clock p.m. a.m. And around 10 degrees here, and then it's so cold time, and I am here with my subject of the presentation. I don't know if you try, you can put to see, but in my mouth say the brain. This is a model, the plastic model of the brain, but I would like to show you the possibility to try and to separate the parts. In my talk today, I explain about this, and they explain about a little bit about our things and the subjects, the most complicated complex system. And then sometimes we talk about the brain, and talk about the brain and not separated this. But today I can put it before this and talk about the cortex. Sometimes we talk about this, and the definitions about this in the books talk about the two hemispheres, and then we can separate this in two parts. Or we can show the complex part when stay the neuron cells. When you talk this, we explain sometimes about the history of the man, and then, but when I start my studies about this subject, I am not alone, and then I would like to show you the group when I made the part in Brazil. I work in an institute. At the moment, I'm head of one institute in Brazil, but not it's about the brain. This is about the engineer. I careful some courses of the engineer and apply the subjects. But in postgraduate, I am member of the one group, and calling the 105 group science. This group we have some research, and these researches stay in some parts of the world. We're not in just in one part of the Brazil or the other things. And this, if you have a curiosity about our group, you can access the QR code or just look in Google 105 group science, and you can verify our group and our publications around the more than 30 years. This is a web page of the group. You can see this page and verify the publications. Because my talk today, I would like to show you the evolution of our words, our papers, our job, our explanation about the brain during the years. Not just one work, is there's a lot works and a lot of tools you can use it to analyze the brain. For the instance, I would like to show so many informations, but I don't read these informations. I after this talk, I can send these slides for the organizers, and then you can access this information. But what do I think about this? If you look at the information about the brain during the years, you can see the evolution is so slow. We have so many informations. The information is so important, but during years, we have no evolution about this information. Because if you try to compare it, our history in the during the years, you can look the slow parts of the discussions, the philosophical discussions about the brain, but not so concrete. And then you try to simulate the best model and the best situations for the brain, but you can slowly. But this slowly is sufficiently for the moment. I think you can evolve along the years. This is natural, but at the moment we have so many informations, accumulated informations, but not the completed explain. This information not completely explain the system. If you try put together all the informations, you cannot reproduce the brain completely. And this is good for the researchers, because we have a future of the works to make. And then when we decided work with the brain, with the networks and simulated the neurons in these networks, we studied the networks, studied the properties of the networks, and after this we can study the terms and insert in our models these terms. One of the most important terms we work at the moment and the past and maybe the future too is the plasticity, because our brain is not static and it change a lot at the moment. In seconds, our brain change. Our brain, when we start this talking, and now it's completely different in terms of the connections. After when you receive a external stimulation, we can change the connections and change this completely the informations, tools, the ways. And then because of this, I talk a little bit about this evolution. When we pay attention about the brain is not static, we talk about the so many years ago. But when you talk about the informations about the Rebel rules plasticity, we can talk about the years ago. And then we look at this and insert in our models the possibility of this change, of these new connections, and look at what happens in your brain if this occurs. In 2017, we published one important paper about this, and it showed we prove what's the Rebel rules real made in our brain, and compared this in the health brain and the disease brain. I show this more in after slides, but if you like to show the main explications for TV, we have one explanation in YouTube. The voice in Portuguese, but you can put the legend and then you can read the subjects. And then let me show about the models and the evolution of the models. Sometimes when we start the talks, the main question is how model is the best to use. This is completely this question is completely outside of the subjects, because it depend of way. What do you have? What do you want with your simulations? Because it is a simulation, yeah. We can use models to simulate the neurons, like Hodgkin-Huxley. This is one model so many accepted for the community because of the influence of the channels, and it's basically one capacitive model, but you can insert in this the channels. We can simulate your network using cellular automata. It's so simple, but not simplest model. You can use the Reed-Marsh-Rose model. You can use it integrated and firing. You can use maps to simulate your neurons. But the important in this case, it's not the name of the model. It's what's happening in this model. This model simulated just one neuron. And then you need to put the set of the equations to simulate so many neurons. If you start with one neurons, okay, you can look for any models and try different models. But if you put in your network so many neurons, and then you pay attention, the careful, because the number of the equations you use to simulate just one neuron. Look at this. This is one example of the paper, the Hodgkin-Huxley, when this is work, the set of the work with the Alan Hodgkin and Andrew Huxley received the Nobel Prize. You can look for 4 equations just in this part, just in this example, but you can have more equations to simulate the channels inside of these 3 equations. And you can insert other terms of inside of this equation. And then you need to be careful about the computational properties and the power of the computation, because you need a computer to simulate these networks. Other thing it's important in this question is what's happening if you use Hodgkin-Huxley or cellular automata or anything model. Every time you can look for spikes and for bursts. You can look at the spikes separated, or you can look at the set of the spikes inside of the period, is you call in the burst. Some models, their behavior, you can observe the behavior of the spikes, and some models just this the bursts, and in some models you can observe buffs. And then you need to decide what you need observe in your model. If you like just to observe the spikes, you can use one model. If you observe the burst, if you can try another model. And if you observe 2 buffs, you can use another model. Before this, let me show. I would like to talk a little bit about the structure. And then we have one big structure inside of our brain, and you need to decide about this, because when you start your studies about the brain, you receive a lot of information about the physiology. And then I think the first information you can look in the books, for example, or the papers is our brain is separated in lobes, and these lobes can be connected. And these connections can be inhibitor or excitator. And this inhibitor and the excitator is inside of the another connections, calling the chemical connections and electrical connections. And then you go back in your model and you need to insert first. You coupling your neurons, and this coupling can be electrical or chemical. After this, you decide what's the percentage, what is the properties of the connections can in between the neurons can be excitator or inhibitor. And then you put this to simulate it. When you transform the physiological connections, the interpretation of the physiological brain in numbers, we use the metrics. And then these metrics can be connected, can be like this, sparse metrics. You have a no connections 0 or connections 1, and can simulate this. For curiosity, if you have a curiosity about this first stage of the structure, you can visit this paper, this like Physica A. I think this is a clear of these connections, the way when how to connect your neurons between neurons, like a connection, direct connections or no connections, shortcut connections, and bidirectional or no bidirectional connection. After this, you can observe what's happened about the data. Sometimes you just made a simulation and observe the behavior about the spikes or about the bursts. This is a first stage when you learn about the neurons, learn about the network, and the next step is use the data. Sometimes we try to use the human data. This is evolution of the work. You try to simulate what's the real we have, but not so easy to looking on the free data about the brain. You need to look and maybe the connections between the experimental researches, and then you can access this data. But on the internet, you can look at so many works about the brains of the animals, and then you can use this. I think the most important is the C. elegans. You can look in this on the internet and receive massive data about this. But we have informations about the monkeys or and sometimes the human informations. The scanner, one researcher present like 23 years of the evolution, the metrics, connection metrics of the cat, and then this is a good possibility of training your model. In these 4 works, I show a little bit about how the interpretations can be for inserting your model. And then we can use the interpretations about if you have strong connections, not so strong, like a medial connections, wake connections. And if you have no information about these connections between the areas inside of the brain, like in this first, you can look at the connections between the visual part of our brain, the visual parts of the connections, auditor, somatomotor, or frontolimbic. This is a good training about the metrics. If you try a little bit evolution of this work, you can separate into inside the visual areas another small areas, and then connection, insert connections in these possibilities. You can try about the percentage, about the influence of the number of the connections, but you can try to the number of the fibers connections. Depends our decisions depend of the interpretation, the physiological interpretation you want. Like in first and second work here, in publishing CSF in the case, we can see just the interpretation of the behavior, but the non-linear interpretation, the complex system. And they don't put interpretation, physiological interpretation. In the second, the third and fourth works here, we need to insert another things, and for the physiological interpretation, we need to insert the new interpretation of the connections, like a percentage or like a number of the fibers. If you decide for the model, what's the representation of your neurons. After this, you decide what's the simulations, the network you can use in your work. And then you can insert in your simulation so many things or many important considerations. And then you can simulate it, our model, your model, and interpret what's happening. In this work, I put here this work because I like so much this, because this is one of the first papers we really show the plasticity and what's happening if plasticity is introduced in our model. And then the main conclusion, our brain is not static. And what's the title traduced for us? Spike time dependent plasticity is a name of the type of the plasticity is inserted in our model. And this type of the plasticity is so present in our brain, the real present plasticity in our brain, induces the non-trivial topology in our brain. And then we start our model. We put the equations. This is our Hodgkin-Huxley model, like the observation here. We have a capacitivity equation, and then we have channels. We insert in this model the plasticity, and the insert the rules of the plasticity to simulate. And then after the simulation, when you use the Rebben rules, when you evolution, we observe the time series of this evolution, the temporal series, and then we observe when insert the plasticities, our network change, and this change is dependent of the frequency of the neurons. This is a most the important thing, because the non-trivial topology is induced in our brain, but you can consider the rubs inside of the models too. And then this is evolution of the model. And then the evolution, I pay attention for the most important properties of the mammalian brain. It's about the no-static information, the no-static brain, and this is a present of the nature. And then I put here just for the information. And we show a little bit of the evolution. After this, we can analyze what's happening about the influence of the topology in the flow of the information in this model. We insert the delay feedback to check what's happening if you insert in your routines like a inhibitor or excitator drugs, and you check what's the alteration in the connectivities the brain happen if you have a plasticity and the synaptic delay inside of the synaptics in our brain. You can observe in these publications the evolution of the model when you have a Hodgkin-Huxley simulated for the dynamic of the neurons, and you can insert the plasticity in this model. And then we can observe what's happening if you have an autopsy. The autopsy, it's I think, when you have a spike and this reconnection inside of the network is around the same neuron, and then we observe what's happening. This is affected the rare effect, but it occurs in our brain. And then you check the B stability firing patterns in this neural network model. And then we check the influence of the autopsy. In our research, all the papers, we can analyze what's happening about the synchronization. Professor Hilda talked about the synchronization a little bit in our talk, and I think it's so many persons today and in another days talk about the synchronization, because this is so many used in our area. And the synchronization, it's one tool, the important tool for our works too. And then we check the synchronization and try to compare what's happening if you have an autopsy or not have an autopsy. In this specific work, you use just the chemical synapses, because when you use the expressions for the autopsy, you can insert just inside of the inhibitor or excitator connections, and then we insert this in chemical synapses, because this is the work is just localized inside the chemical synapses. And then we not so happy with the results, and then we try a lot of things about the influences of this delay inside of the conductance. Because if you remember the Hodgkin-Huxley model, for example, we have a capacitivity equation, and then we have a channel simulated in another equations. But when you look in this model, the influence of the conductance, it's a lot. And then when you analyze the evolution of the models, when you insert the drugs, we analyze what's happening with this conductance. And in experimental works, at the moment, so many researchers look for these influences. And then if you try, if you start your work now, or you look for what's the good way today, I think the conductance, it's a best way. And then we'll observe what's happening about the synchronization between the areas, inside the areas too. We observe the complex influence of the inhibitory synapses when you insert these synapses inside of our model. And try what's happening if you put noise inside of this model, because sometimes we observe the networks, we observe what's happening about the synchronization, we try to explain diseases like Alzheimer or Parkinson or some lesions, but in the real life, like noises, the sounds, or external stimulus, what's happening in our brain if this type of the influence is inserted. And then we try and publish this paper, noise-induced continuous and non-continuous transition in neuron inter-spike internal range. Because we observe just the modification when you look for inter-spike intervals. In the evolution, this is a good way to start the work like this. When we is compared like a theory of the temperature, we have a first law, second law, and then you go back to zero law. Here we study a long of time and inserting our models type of the connections and the comparated after this what's happening. But along the years, we have one thing about this. What's the best properties we need to use to start our works? And then we start the works to try these tests. And we take the human data. We receive the real data from our experimental collaborators. And then we analyze the network properties in health data brain and the Alzheimer data brain. And then we comparated what's happened and try to approximate what's happening in one and another. The main conclusion about this in our results, we can observe the properties, the statistic properties like assortativity, but not sometimes, not real times. So many researchers use this type of the tools, but it is a important tools to observe in our models. Our our results indicate about the small world networks is the best to start our simulations, and they check all the properties, the statistic properties to affirm these results. But among the neurons, the communication of the neurons, we check the continuous of this work in these two works. In the first, we use a network model. And they study what's happening in the phase synchronization of this human matrix. And after this, we study the synchronous behavior based on a cortical connection in human brain. And then you use the real interpretations and the physiological interpretations of the brain. Here we have just the visual information about the human data. And to close this part of the model, I don't show you the conclusion of the works, the papers, because you can look at this paper pass to paste after this talk. But I would like to resume our more than 30 years of the evolution of the models. Now we have some networks, we have tests, a lot of tests about the burst spikes and the experimental mini brains now. We have a simulation, a lot of simulations and in different types of the models. Oh, just, and the, oops.
Yeah, I think you should now start winding up.
Yes, and then just to finish, we can receive after this, but this is a resume of my talk. We have so many informations, and we have so many possibility about the tools to analyze your network. I would like to give acknowledge everyone to watch my talk. I like too many. I like so many the look. Let me try to here. Oh yes, and go back.
So thank you, Kelly. Any questions? Anybody? Yeah.
Hi, nice talk. What do you think about the brain fluid dynamics? Is it possible to merge the fluid mechanical information about the system with the neuronal connection architecture to basically provide some kind of constraints or boundary conditions on how the connections might work? For example, like the fluid mechanics is important to carry the nutrients or to take the waste matter away from the brain, right? And that is connected to some clinical conditions like Alzheimer's. So are there groups who are working in the interface of these two fields?
Yes, we have some works about this influence. We start our works in this way when we started our connections between the research called the Murilo Batista. He works in Aberdeen University, and he works so many with the transition of the information. Yes, and then for these models, we need to consider it in our subjects the fluid contacts, and then we have some works, and I think the evolution of the models, it's not we cannot abandon these considerations, and then it's important.
Anyone else? If not, thank you very much, Kelly, and thanks for being here at this. I don't know what time it is for you. So this is the end of the session. So let me call the session to a close. Thank you so much.
Okay, thank you. Thank you.